Hello and welcome to Beginnerology. Today we will do a thing which is taught in many classes, mainly class 12, about structure of chromosome, structure of DNA and how DNA replicates. DNA is a molecule whose structure was discovered by Watson and Crick in 1953. They have predicted the DNA structure which is sent in Nature as a small paper. Later on, Michelson and Stull in 1958 proved that what Watson and Crick proposed the structure is correct. And there we are, which is known as semi-conservative replication of DNA. So coming to it, what is DNA? We have tried to make a structure of DNA which appears to be two chains which is joined double helically. This long chain of DNA, this makes one chromosome. One chromosome has a very long DNA. The two chain is bonded by just hydrogen bond. The moment helicase goes through it, it opens up. Now how this DNA is in chromosome? So this is, suppose the blue one is DNA chain, which in chromosome is around a histone. A long chain of DNA, a chromosome made up of single DNA, long chain of DNA which is highly supercoiled. Basic histone protein which make a round globular shape of H1A, B, C, D. 4 histone makes pair and 8 histone makes a globular thing around which DNA is coiled 2/3/4 chain. And then in the last, the H1A attaches it to make it attachment, and then the next one. So suppose this is histone protein and this is DNA which goes around it. So this is one histone protein, HA1 attaches here, then another one. So this we call nucleosome. So nucleosome with DNA attached to it. So this is histone protein, 8 histone makes this octamer of nucleosome around which DNA is attached, and then this whole thing is recoiled to make a chromosome. So this I have shown two color. So the orange change is a new one which is prepared in the old chain, the white chain. And so this is semi-conservative DNA. This DNA has joined in between, and that is done by nucleotides. So DNA is nothing but polynucleotide. Of that, each nucleotide, each nitrogenous base such as A combined with T with a double hydrogen chain, GC with a triple hydrogen chain, AT with a double bond. So there is no bond as such in between. So they are attracted to each other, and so when helicase goes through it, it opens up very quickly. So this is sort of a small model of DNA. Now how to make this DNA? So we have done a video about protein synthesis where I have shown how to make DNA. So I am repeating here. Look at the paper. This is a A4 card paper and I am making it half longitudinally and taking the pencil to make a template. You may get template given below. After that, we are using scissor to cut this. So if you cut few such things, then we can join these with the help of sellotape into say 3 of them. Similarly, we can join 3 more. So we can then make it crisscross to make a DNA. So this sort of become a DNA chain. To make DNA, we need nitrogenous base like ATGC. So what we are doing is in the same template. So we are following this template. DNA, which is purine is A, pyrimidine is T, purine is G, guanine, and pyrimidine is cytosine. So here in this DNA template, suppose this is 5 prime, this will end in 3 prime. Other chain will be 3 prime, which ends in 5 prime. So here is one DNA, this 2 chain. So arbitrarily we can join. This corresponds T. So opposite will be A. This is A. Opposite will be T. This is C. Opposite will be G. And so on and so forth. So this is one DNA strand. So DNA strand which is coding here is in the form of this. So there is one chain which opens up and makes new DNA is we call template strand. So 3 prime 5 prime DNA strand which opens up and makes new DNA we call template strand, and the other chain which is countering that we call coding strand. So coding strand, remember coding strand does not code. Coding strand is similar to next one. So coming to it, here DNA opens up. So opening DNA, there is a special enzyme known as helicase, and one enzyme called helicase which goes through it, opening the DNA strand. So this DNA strand opens up, helicase goes through it. The moment it goes through it, the attraction between A and T G and C, which is nothing but hydrogen bond, opens up. So DNA polymerase comes here and start making new DNA. DNA polymerase cannot work de novo. So it need another enzyme known as primase. So primase comes here and start making a small RNA chain, which is also will work similarly to 5 prime 3 prime direction. So initially primase comes, make a small RNA chain which is also 5 prime to 3 prime, which just opposite the DNA will be, as DNA is TTA, it will be AAU because this is RNA. And after that DNA polymerase tree will work itself. DNA polymerase tree will start working, start joining the new DNA chain. So here, after the small RNA chain, DNA will keep on making it according to the template. So if it is C, opposite will be G, G opposite will be C, C opposite will be G, G opposite will be C, A opposite will be T. Yes, in this case it will become T because it is DNA. C opposite will be G. So this way this part is opening up. Helicase goes further and further opens up. This is attached. So up to this is known as leading strand, it's leading here. Now helicase goes further. Whereas what happens to lagging strand, the other strand? This happens strangely a little different. This, everyone tried to find DNA polymerase which work 3 to 5, because what happens to the other chain? When they are trying to find what happens to the other chain, a brilliant scientist named Okazaki got an idea. So Okazaki came to an idea of making a small part of RNA with the help of primase. So this is G, opposite will be C, C opposite will be G, and G opposite will be C again. So this is an RNA. Then they make the other part of DNA. Here again one DNA polymerase comes and makes this chain works 5 to 3 only. So this is 5 prime, this is 3 prime. Here the DNA polymerase make T opposite that, it will be A, not U, A opposite will be T, T opposite will be, A opposite will be T again. So I am writing here C opposite will be G, C, C opposite the G, then T opposite A opposite T, opposite T. There is a small fragment of DNA making in the lagging strand. We call lagging because it takes a little bit slow compared to leading strand. That Okazaki found. What he did is he start growing E. coli which has generation time only 20 minutes. So within the 20 minutes the whole cell divides. So even in less than 20 minutes, in 15-16 minutes DNA duplicates. So he has taken a pulse technique. He started taking out new DNA in, say, in 1 minute, 2 minutes, 4 minutes, 6 minutes, 8 minutes, even in seconds. He found lots of DNA. So one DNA he found keep on elongating normally in 5 to 3 prime direction, he has said as leading strand. Another one was happening with small fragments. He named as lagging strand, and later on in his name we now call these small fragments as Okazaki fragment. This way this helicase keep on opening the chain and new DNA keep on extending, we call, thanks to DNA polymerase. So this A opposite T, G opposite will be C, so as helicase goes further, leading strand keep on leading and joining it. So G opposite will be C. Meanwhile, what is happening in the other chain? The other chain, another small RNA primer is being made by primase. Suppose a small primer here. And instead of A opposite in DNA happens to be T, but in this case it's RNA, so it will be U. This is 5 prime to 3 prime, so here this is again a small fragment of DNA which is new one. So helicase done the leading strand up to this, whereas lagging strand is still lagging behind. Helicase goes further and opens up DNA. So for our case, this is a small DNA, so this opens up. Helicase goes further and opens up DNA. And so here leading strand go further. So this is 5 prime, so here will be the end in 3 prime. Meanwhile, what happens in the Okazaki part, the other chain? So here primase make little more RNA chain. The joining is done all by ligase. Ligase is also known as molecular adhesive which joins DNA. So the cellotape is working as ligase. We got here TAAU RNA primer. After this primer, DNA polymerase start making guanine opposite cytosine, adenine opposite thymine T, adenine again thymine T, G here become C, C here become G, G here becomes C. So these small fragment of DNA, that is Okazaki fragment, completes. Now DNA polymerase 1 comes to read the proof. So DNA polymerase 1 when goes through it, wherever there are mismatch, it cuts and makes it. So the moment it comes to UAA, that is RNA, it simply removes this RNA and makes DNA. So against A, instead of U, in this case it makes T. This is their work of proofreading. T opposite A, T opposite A, and this remains as 5'. Same way DNA polymerase 1 goes through Okazaki fragment and remove the RNA wherever it gets. Wherever it's mismatch, such as this, it removes it and makes a DNA. Still there are some part of RNA this way, RNA is being removed and DNA is joined thanks to DNA polymerase 1. Roman 1 or 3 is the way discovered. Which discovered first we have named as 1, but later on we came to DNA polymerase 3 works for actual DNA polymerase, and DNA polymerase 1 only do proofreading after DNA polymerase 3 has worked. We got two DNA, one from leading strand, another from lagging strand, and that is semi-conservative replication. So the brilliance of Okazaki is to try to find out the DNA replication, whether it happens by DNA polymerase 5 to 3 only, then what happens. So he got that thing. And Meserson and Stull has done strange experiment of N14, N15 medium, where they have grown E. coli in nitrogen 15 medium for many generations, and so that all DNA of E. coli becomes N15 heavy, and that when they centrifuge it becomes, in cesium chloride medium it goes further out as it is heavier. So heavy DNA makes a chain, but light DNA makes a chain which is further up as it's less heavier. If it is semi-conservative replication, in that case this is, one is N15 and one is N14, so it's a middle, it should give a middle chain which they have predicted, and that is what is found, and that proves that Watson-Crick's proposal of the DNA double helix chain is correct. So here DNA is double helical, where new chain is the black one and old chain is the white one. DNA replication we have discussed. DNA replication starts in bacteria which has a circular DNA. In some point we call origin of replication, and basically DNA replicates and become 2 DNA, and after that it again opens up and makes 4 DNA, and so on and so forth, as we know in PCR machine. PCR machine, we have found a strange DNA polymerase which we got from Thermus aquaticus bacteria, which can grow as high as 100 degrees Celsius. So we got a DNA polymerase which can work up to 96 degrees Celsius without any hindrance. thanks to Yellowstone National Park where we found this. And the person who found it never thought what he discovered. He got Nobel Prize. He was surprised. He said, why? I just discovered one enzyme. But he opened up a full area to work. Now we got a machine where we put any DNA, even nanogram DNA from fingerprint or from anywhere, any cell where a murder happened, murder victim's skin or anything is there or blood. So from there we got DNA nanogram. We just put in a machine known as PCR machine which do polymerase chain reaction, and DNA is heated up, annihilated, and so in case of helicase which opens up DNA, in this case heat opens up to DNA. Discovery of Taq polymerase make it possible so that we can put DNA in a machine and machine breaks it into two, and that two DNA chain, and due to ATP, GTP, CTP and TTP already given there, this new DNA is formed by Taq polymerase, and so we got 1 to 2, and then again it heated up, again annihilation, separate out happened, again new DNA forms. So in this way PCR machine within few hours can make nanogram of DNA into microgram or milligram, so that we can do experiment about DNA fingerprinting, and this way we can get the culprit. So amazing discovery. So here we are concluding here that this way DNA can replicate semi-conservatively, which was proposed by Watson and Crick about the model. I hope now you will understand how DNA replicates. If not, please write to me. I will always respond. My email ID is given there, jparuk@gmail.com, as well as vigyanclub@gmail.com. And class 12 aspirants, class 12 exam is very nearby, so go through my video. I think it will explain DNA replication much better. Like, comment and subscribe. Thanks a lot.